Can everybody see that? Do I need to make that bigger? Too big. No, not too bad. So what I did was I downloaded that gene list. So these are genes that were differentially expressed in at least, I think it was at least one of these stage 3 versus either stage 2 or stage 1. You can see the specificity. So we had 24 total. This gene in particular, depth domain containing 1, was in 21 out of 24 studies, or 87% of my studies. If you look on the right, it gives you the fold change for all of those studies where it was differentially expressed. And as you can see, they agree quite a bit, right? If it's down, usually it was down in all the studies. If it was up in most of those. What I did was I basically took that list and found the median activity. So I took the median fold change for all of those different studies, number 1. So basically that's kind of giving me an average fold change that I know this gene is important in stage 3 breast cancer. Yeah.
I'm just curious, do they specify in the studies if it is coming from a comparison between cells coming from patients or cell lines that are usually representative for a certain state of...
We took from tumors from people. So these were all tumor.
I mean, is it specified somewhere? Because a lot of studies actually just take cell lines that they say this cell line is a representative of that stage, and sometimes it just compares cell lines.
In each one of these studies, you can actually go in and get all the study details.
Where do you get it? I mean, yeah. I'm sorry,
So,
but this is a very important detail when you work on cancer.
Totally.
You know, if the cells are coming from patients, so not cultured for
Totally.
a long time.
And at this point, I would say probably there might be a mix of that, and that's okay.
Yeah, that's why.
Right?
Yeah. It's just good to know and to be able to exclude the ones that you want to exclude.
Sure. Absolutely. If you wanted to, for this thing, we were really concerned with like making sure that it was, tumor versus, cell line. We just wanted stage 3 versus something else, right? And I know out of the 24 samples, I'm sure some of those were cell lines. But if this gene is differentially expressed in 21 out of 24 of those, regardless of if it's tumor or cell line, it's probably important in stage 3 breast cancer. Trust me. Wait till you see the analysis. Okay? So what I did was I basically required that, and I got a lot of genes when I actually did this analysis. So what I required is that this gene, whatever gene I picked for my final analysis or for my final gene list, that I wanted it in at least half of the 24. So it had to be in 13 out of 24 studies differentially expressed. When I did that, I ended up with 970 genes. Okay? All right, close that. Just make sure, sometimes it shuts off. I just got to open it. Yeah.
Is there a way where you can input your own RNA-seq data into NextBio?
Yes. So this is what I'm going to do right now.
Okay.
So I made that list. You can now put this back into NextBio. So you basically go to import your data. Takes a while. That's the best thing about this, is I can take any gene list I want, I can throw it into this, and it'll compare it to 20,000 different gene lists out there. What happens is then it ranks them based on its correlation to those gene lists. So what happens at the top of those lists is basically telling you what kind of gene list you have, what kind of processes it's related to. So I can choose my project, breast cancer. I can name this study. I'll just go test. Appropriate data type. This is RNA expression. Select experiment for organism. This was all Homo sapiens. Then I can choose the file. And I've got it already set up. Go next step. This is pretty easy to, and this is pretty similar to most bioinformatics software or database programs out there. It's pretty easy to upload your data. What they want is they want you to tell you have to tell the software what column contains your gene or probe set ID. some kind of unique identifier for whatever you're looking at. And also, we also have the mean activity, which is our fold change, and I can tell that the fold change. And that's important information. It's not only important, and I want to stress this, it's not only important to know if your gene is differentially expressed, but what direction it goes. Does anybody here use the David bioinformatics software? It doesn't take any of that. To that, it's just like, hey, there's a gene. I'm just going to text search this, But, if you don't know which direction, the direction the gene is going is very, very important. And if your database, your bioinformatics tools don't take advantage of that, then I think you're getting shortchanged. All right, so we can put our fold change in here. If I had a p-value, remember we just generated this gene list, but if you were putting your RNA-seq data, you would put your fold change information in there as well. Okay, go next step. View and summit. There was only one gene that it didn't recognize, and that was probably our column header. And I can hit next step. And you can put any tags, and this is nice, this will allow you, if you want to go back and go, I want to find all my data sets that had this keyword. I could put in breast cancer here or whatever I wanted. Stage 3. Finish. Go to my studies. So this is going to take a while, but in the meantime, though, I have already done this. So here is this list. Open it up. So stage 3 versus stage 1 and 2. Again, medium activity. This was built on 24 gene lists looking at grade 3 versus lower grades of breast cancer. And again, it's about 970 genes with fold change information. Okay. Again, this is showing me how many genes I have. I can click on this red. This is the quick view. So this basically takes my gene list and uses this as the benchmark for the software. So all the software is going to be acting on my gene list now. Again, we have our gene list right here, or genes. You can sort this any way you want. You can download this. You can share this information with other people that have the same program. Now, this is what I really like about this program. So we've created this data set. You can actually look, you can hit curated studies. And of course, my gene list is very similar to a lot of breast cancer data sets out there. In fact, there's a ton. I can also, and this is key, especially if you are analyzing tissue that contains lots of different cell types, I can go to Body Atlas. What this is going to tell me are all of those tissues where my gene list is either positively or negatively correlated based on the direction of my genes. And as you can see, these are all negative here. These are the immune system, blood cells, which is kind of funny. What I find is that when I analyze these gene expression data sets, if the cancer is going to get worse, somehow you have to impair the immune system. So a lot of times what you'll see is a negative correlation with the immune system that goes with it. Now, of course, I'm getting a positive correlation, which is the green color, with my breast tissue. We can actually go into certain cell types. Say you're looking at a particular cancer, you've got a list of genes that are differentially regulated. You might be able to tell what type of cancer cell that came from.
This is based on a database of immunohistochemistry or RNA?
So this is based on all of their genomic data that they have in here. They're curated data sets. It's based on 20,000 different data sets out there. So basically what they have is they've already done the statistics on all those data sets. They have all their comparisons. They generate gene lists. And then you basically put your gene list in, and it compares the two. How many are similar between this other gene list? Not only that, but is the direction of gene expression the same, inverse, or is it positively correlated or negatively correlated? Does everybody understand that? Okay. We can go to Disease Atlas, and obviously cancer will show up. But there's some surprises here. Actually, when I did this, a lot of immune stuff comes up, which you wouldn't necessarily expect. So you see the severe acute respiratory syndrome, and you get a lot of respiratory disorders kind of show up with breast cancer because it's both epithelial-derived cancers. But this is SARS, and what they're showing is there's a positive correlation with SARS infection. And what happens with viruses, they also suppress the immune system. There might be something similar going on. Okay, skin shows up. You can do pharmacoatlas. So based on my gene list, it's going to try to find drugs and chemicals that are either known to inhibit my genes on my gene list or activate them. And I will tell you right now that this is positively correlated. This is I think this is a known, positively correlated, yeah. This is I think they're using this as a cancer therapy. And here, I think this is known, it's a carcinogen, I believe. Oh, wait, no, sorry. This is actually, they've used this as a anti-cancer therapy here because it's negatively correlated. This positive correlated, this daidzein, and I looked this up, it's kind of a derivative of soy. If you look at this, this is very much like estrogen. Yeah, breast cancer, estrogen. What's that?
And the estradiol.
Yeah. Yep. So again, not only will this allow you to compare your gene list, but it will also tell you potential drugs that might actually influence your system. And knockdown atlas. And we can also go to pathway analysis. I'm kind of running out of time, I'm sorry, so I'm going to kind of speed it up here. And this is kind of your basic bioinformatics analysis on the gene list that I just put in. So I put this gene list, what it's telling me are all of those things that are overrepresented in my gene list. That if I look here, genes involved in cell cycle, I'm getting a lot of similarity between my gene list. Not only that, but the odds of me getting all of these genes associated with cell cycle are like 2 times 10 to the negative 80th. That doesn't happen by random chance. And when you see p-values, and this is why this meta-analysis works so well, is I've noticed when you take a large group of studies that are looking at the same thing, and you take pieces out of all of those and put them together, it's actually stronger than any individual study itself. Anybody heard of Sir Francis Galton and the Wisdom of Crowds, the bull? It's kind of the same theory. No one's heard of that? Oh. By the time I leave campus, everybody's going to know Sir Francis Galton. Well, he was Darwin's cousin, and basically he went to this county fair, English county fair in like the early 1900s, and they were auctioning off a bull. And basically everybody was guessing, so that if you guess the person that guessed the closest way to the meat that was processed from that bull got that meat. So there was like 800 people that guessed. No one got the exact answer right, but when you actually took the average of everybody's guesses, it was the exact way to the bull. We can do the similar thing in biology, I'm telling you. It works. It really does work. But again, this gives you an idea, and you could go in here and you could download any of those. Any of these groups of genes associated with any of these. Spindle, DNA repair, kinetic core. All right. So this is kind of the NextBio. Any questions on NextBio right now? Okay. So this is a good way to generate your gene list without having to download an actual study and knowing anything about programming and mathematics and all that good stuff, right? Now what you can do is, so this kind of this allows you to generate your gene list. The kind of the Cadillac of all bioinformatics programs out there is probably IPA. Anybody here work on IPA besides you? Oh, we got 2, yeah. We got 2 people. All right.